So this is a presentation about some results from an interlaboratory study of the force panel, which is a SNP panel that can be used for different forensic purposes. So these results presentations were presented at the 12th Haploid Markers meeting in Budapest, Hungary, earlier this year in May, with the title Advancing Forensic SNP Typing, Insights from an Interlaboratory Study of the Force Panel. So this interlaboratory study was conducted with 15 different labs contributing around the world. So first, some introduction to what this project originated from. So SNP markers have been around for many years within forensics, but not been given that much attention. Instead, STR markers had been sort of the golden standard DNA marker. However, more recently, SNPs has been of more interest due to different advantages that these markers have, both for forensic, kinship, and missing person analysis cases. For example, they are very suitable for analysis of using high throughput sequencing technologies like next generation sequencing or massive parallel sequencing methods. One benefit with SNPs compared to STRs is that since it's the variation is only one nucleotide of length, you can create very short fragments or amplicons that can be analyzed, which is beneficial when it comes to analysis of degraded DNA samples or other bad forensic quality samples. In the human genome, there are also a large number of SNPs to choose from, which is also beneficial, of course. For kinship situation and kinship cases, the lower mutation rate compared to STR markers has also an advantage when it comes to interpretation of close relationships like parent-child or any other similar relationship. And also, relative to STR markers, SNP markers are quite easy to interpret when it comes to genotype interpretation, since there are only maximum 4 different variants or alleles per marker to sort of choose from, and the possibility to do mapping and genotype calling with only one nucleotide is also a lot of easier compared to STR marker mapping and genotype calling. However, as I said before, since these markers have been around, there has not been that much effort to sort of unify which SNPs to be analyzed within forensics. So a lot of different panels around, smaller panels with variants, SNP panel designs and choices. So there is a need for standardization of forensic SNP markers. And what we, when we started this project many years ago, we thought that many different laboratories have different preferences when it comes to experiences and when it comes to like library preparation methods and techniques, and when it comes to sequencing instrument, if they use a MiSeq or an ION S5 instrument, for example, and also the bioinformatic tools and know-how. So what we wanted to see when we started this project 2, 3, 4 years ago, we want to see if there was a possibility to, instead of making sort of one solution, making a suggestion of a set of SNP markers that could be analyzed with different assays and different sequencing technologies based on each lab's possibilities and old preferences. So the question was, if it was possible to customize sort of a complete workflow from DNA, going from DNA to the genotype for a SNP marker panel of this size, around 5,000 markers. So then we came up with a suggestion of a set of SNPs to be included. These are in detail described in the paper from 2021. We named the panel Force panel, consists of around 5,500 SNPs. The SNPs has been chosen for different various forensic applications like identity and kinship testing, but also ancestry, phenotypic predictions, and things like that. And one thing that we wanted to focus on was that we did not want to select and include novel SNPs, but rather SNPs that has been around some years and also been studied before, but also that they are available to analyze in other SNP panels. It could be a SNP chip, it could be another dedicated panels like from Verigen or QIAGEN or Thermo Fisher. And the benefit with this is that it would be beneficial when it comes to cross comparisons and validations of the panel and things like that. For example, the situation could be that one lab uses another assay to type a set of identity formatted SNPs, and to aid cross comparison, of course, if your own lab have 4 SNPs, it's very useful if it's possible to compare different SNP profiles from various kits. So therefore, when we designed this and selected the SNPs for the forensic panel, we looked at what was existing, and for example, for the kinship SNPs, all the kinship SNPs that are included in the Force panel are also included in the major SNP chips, like the GSA chip, GSH chip, OmniExpress chip, etc. And for example, the ancestry SNPs are already included in other ancestry-focused panel, like the Precision ID ancestry panel from Thermo Fisher, the Visage panel from the Visage project, or like the forensic DNA signature prep kit from Verigen or QIAGEN. So what about the 4 SNPs? I've already shown that they are selected for various different applications. Like, for example, there are around 240 SNPs that can be used for ancestry predictions, like from where in the world an unknown individual originated from genetically at the sort of meta-population level. And there are SNPs included that can be used to predict eye color, hair color, and skin color. And these SNPs are the ones that have earlier been developed and studied by Manfred Kaiser and Susan Walls and their colleagues. But the majority of the SNPs included in this Force panel are kinship and identity SNPs. And for the kinship SNPs, the amount or the number of SNPs included is from which they're available to sort of infer and predict up to 5th degrees relationship. And there are also some X chromosomal SNPs and Y chromosomal SNPs included. The Y chromosomal SNPs can be used to sort of detect paternal lineage comparisons between male individuals, but it can also be used to predict biogeographical ancestry on the paternal lineage. And these SNPs that we have included are also the ones that have earlier been studied and developed and designed by Arvin Rolf and Manfred Kaiser's group. So, as I said before, the idea behind this project, or one of the ideas, was instead of proposing a specific assay with a specific SNP panel, we instead wanted to propose a SNP panel that can be used with different assays and technologies to analyze the same set of SNPs. So currently there exist 4 different assays and technologies that can be used to analyze these Force SNP markers. And those different assays are based on various or different kinds of technologies in order to analyze the DNA and the SNP markers. For example, the MyBates from Dayshell Arbor Biosciences uses a hybridization capture technique to capture the fragment containing the SNP of interest. The QIAseq from QIAGEN uses a primer extension capture technique, and then the AmpliSeq from Thermo Fisher and the XGen from Integrated DNA Technologies uses a traditional PCR-based amplicon analysis in order to capture and analyze the SNPs of interest. So when it comes to my baits, if you're interested in that sort of technology and application in this sense, you can read more detailed information in the Force Panel original study from 2021 paper. And if you're interested in the QIAseq, you can read more in the paper from 2023 by Stadig and colleagues, which describe the QIAseq implementation or version in more detail. Just to briefly compare and introduce you to different technologies, like the hybridization capture, they start with the genomic DNA, do fragmentation, add adapters to each of the fragment DNA templates fragment, and then all the fragments that contains the SNP of interest are captured by hybridization capture using probes that are designed to hybridize close to or over the SNP of interest. This means that after the capture step, you only have the fragment containing the SNP of interest, and you amplify these fragments, and then you do traditional sequencing, like NGS sequencing with Illumina or Thermo Fisher sequencing with Ion S5, for example. QIAseq version is quite similar. You start with genomic DNA, you fragment the DNA, you add adapter, but you also add something called UMIs, which is our unique molecular indices, which are unique DNA sequences that are tagged to each individual DNA fragment. And you can use them in the bioinformatics in order to filter and exclude all PCR duplicates. So you, in the genotype calling, only do the genotype calling from individual DNA templates. The targeting and the capture is done using one primer that anneals close to each SNP of interest, and during this amplification, you only end up with your library containing fragments that contains the SNP of interest. And then you sequencing as you sequence the library as you do with the hybridization capture libraries. And the third sort of category of technologies, for which the AmpliSeq and the XGen implementations are formed on, also starts, of course, with genomic DNA, and then uses a traditional PCR amplification workflow where you have a pair of primers for each target. You amplify the regions of interest, and then you add your adapters, amplify the library once again, and then do the sequencing like you do for the other technologies. So that was a little bit of background for the Force SNP project, sort of from its start. What we wanted to test then is our theory to see if it works, that different labs use different technologies, different preferences when it comes to settings and things like that. So still the question is it possible to customize the complete workflow going from the DNA sort of to the genotype for the Force panel, comprising around 5,000 SNP markers? And in this interlab study, as I said from the start, 15 different labs around the world participated, representing a wide difference between variety when it comes to different technologies used. So all these 4 different assays are presented. We also have seen that the different labs use different sequencing instruments. So we have also different sequencing instrument and sequencing reagents present and included in the study. So each lab that participated in the study were given 12 different samples, DNA, to be analyzed. So those 12 samples comprise both of good quality samples like Coriell cell lines and NIST reference samples. There were also a dilution series included, going from 30 picogram of total input DNA up to 10 nanogram of total input, and also some bad quality samples like degraded DNA and humic acid inhibited samples. So each laboratory were given the task to analyze the sample based on their selected assay, sequencing instrument, sequencing strategy, and also based on their own setting and choice, preference, and experience. And in this way, we thought and still think that such an interlab study make it more realistic, because different lab uses different assays, different sequencing instrument, different settings, and therefore useful to sort of make use of the results from the interlab study. However, of course, as we will see later on, it's harder to slightly harder to compare the results due to the fact that they have used different preferences. So here's a table for the different sort of variability for the different parameters, like the choice of assay, the sequencing instrument, sequencing kit, and of course the number of samples that was sequenced together, which also have a high impact on the possibility and the probability to sort of obtain useful genotype data. And what we can see here is that all the different assay types has been represented. We do have also representation from different sequencing instrument and different sequencing kit. And also we do see that there are uses of different cycle numbers for the different labs and protocols. And also there's a variability in the number of samples that were sequenced together. So the genotype calling, the bioinformatics, was done in one lab for consistency. So we used CLC genomic workbench for the bioinformatics. And there were some differences when it comes to tools, bioinformatic tools that can be used. For example, both MyBates and QIAseq, you can exclude and remove all PCR duplicates based on the fact that for the MyBates, you can see the different length of the fragments and remove PCR duplicates. For the QIAseq, all PCR fragments or all reads that contain the same UMIs were merged together to super reads, and also therefore sort of removed the PCR duplicates, while this possibility is not possible for the AmpliSeq and XGen. And the final genotype calling was done in an R script that was sort of built on a Excel spreadsheet that were developed by Kimberly and Yagi. As a threshold we used in the data that result that you will present, that I will present here, we used a coverage threshold of 10x in order to be call the genotype, but also we looked at the allele read frequency, which means sort of the allele balance, in order to separate the heterozygous call from a homozygous call. But also there was a interval between the heterozyte call and homozyte call where there were no call due to sort of no data, not sufficient data to do the calling. So some results. Here is a typical example from one good quality sample. It's the Coriell sample, NA12878. And what we see here is the genotype call rate and the accuracy. So this is for the autosomal SNP markers, a little bit more than 4,000 of those. The green here is the correct genotype obtained for the lab, like the Chi-seq number 1. Since this is a Corel sample, we do know the true genotype, and we can measure and see not only the call rate, but also if it's a correct call or incorrect call. So what we do see is that for each of the different assays, we do in principle get 100% call rate and accuracy, 100% accuracy as well. For the Chi-seq, and we also have representation by the AmpliSeq, which is principle, and XGen and the MyBaits. So each of the different assay works well on good quality samples. However, we do see some marker dropout for some specific labs. For example, this is MyBaits lab. There are only sort of 60% of the SNPs that were called. Everyone, in principle, all of the SNPs that were called were accurate. However, there were a lot of marker dropouts due to the fact that this lab used a micro reagent for the MySeq sequencing, which means that the total number of reads per sample, since they also multiplexed 12 samples in the same run, were not in the same range and comparable to the other settings that the other labs uses. And another similar issue was with one lab with a high level or high proportion of no genotype calls, which most probably was due to poor sequencing reagents quality. However, overall, very good call rates and accuracies across with a 99.9% accuracy on average. However, what we did see were some errors, and this is based on, some of these are based on allele dropout, like you had only observed one of the 2 alleles in the heterozygous for some few instances when it comes to good quality samples. We also saw that there were some complex regions that for some of the sequencing instruments did not produce accurate results due to the mapping, and this could be also a mapping issue. However, these were very few, and since, for example, the AmpliSeq was here tested for the first time, some of the SNPs can be removed prior, for example, implementation for casework due to, for example, complexity and things that make the genotype calling a little bit harder. What we did see, even though for good quality samples, there were on average high coverage, high call rate, and high accuracy, we did see quite large variability when it comes to the underlying coverage data. So we did use a 10x threshold to call the genotype, but of course, looking into more of the coverage, we did see a large variation when it comes to the average number of reads covered for each SNP. And of course, it's hard to compare across all the different assays, because for the Chi-seq and the MyBaits, all the PCR duplicated reads have been removed compared to the AmpliSeq and XGen. However, within each assay, it's possible to compare, and we did see some labs having a lower number of average coverage compared to other labs. And of course, for good quality samples, this may not impact the output or outcome that much. However, if you use and analyze a bad quality samples, this may have a much higher impact on the genotype calling. So here's an example. Another example is more, if we look more on the more complex or forensic-like samples, like a degraded sample. Here's a degraded sample that had a mean fragment length around 200 base pair. We did see some issues with the AmpliSeq, although some of the AmpliSeq labs produces quite high number of correct genotypes. There were also a few that did not produce any calls at all, and also a higher error rate for the PCR amplicon-based assays compared to the other one. And the majority of these errors consist of dropouts, which means we also saw that the design of this AmpliSeq panel, the design of the amplicons is PCR-based. It relies on the fact that you are able to anneal both primers to the same target, which if the primers are too far apart, it will not be able to amplify if there is a degraded DNA sample, like in this case. And the inhibited sample worked well. We used humic acid at the level comparable to what other similar studies have been used. And overall, it looks very nice and good. When we looked at dilution series, we wanted to see if it's possible to produce results even if the level of input or amount of input DNA were sort of low, as you can expect in forensic situations. What we did see here was that a large variability across the different labs. However, we did see both for the QIAseq, the AmpliSeq, and the MyBates, and looking here at the 100 picogram total input, in principle, there are labs that are able to produce almost 95 to 100% call rate with, in principle, 100% accuracies for each of the different assay types, QIAseq, AmpliSeq, and MyBates. So each of the different method worked well. However, due to settings and other characteristics that were sort of chosen, that were made with different labs, we do see a lot of variation, both when it comes to the coverage, sort of from going from no calling up to 90% in this particular case of a genotype called, but there were also labs that had a higher level of incorrect genotype call, and in this example, it's due to dropout of alleles. And of course, here the thresholds and settings comes more into play compared to analyzing and bioinformatically analyzing and you do genotyping for good quality samples. However, it's also important to notice that even though you don't get a full profile, like with samples having not a lower level of input DNA, you did get useful results. So this is an example from the dilution series on the Y chromosomal SNP markers that can be used for haplogroup prediction. So here's a representative sample from one of the labs. You do see that going from 10 nanogram of total input down to 30 picogram of total input, the number of cooled SNPs decreases as expected. However, even though in this particular example, we had around 800 SNPs that were cooled from having good quality and high level of DNA. This lab was only available to cool 66 of the Y SNPs at 30 picogram. But looking at the use of the data, the 66 SNPs did have the same level of resolution when it comes to haplogroup prediction. So even though you don't always get the full profile, it can still be useful due to the fact that the total number of markers included in this panel are of such a high level. And similarly, when it comes to kinship analysis and using the kinship SNPs, It doesn't, even though you don't get the full profile, you can still make useful information out of it. So here's from simulation, and this is simulated based on the kinship informative SNPs for a third-degree relationship, like a first cousin or a great-grandchild relationship. And if you have complete profiles, full profiles, you get a very high level of information. The likelihood ratio is very high compared and well separated compared to unrelatedness between two individuals. And you also see that going down to the light green here, you only representing 25% of the kinship SNPs that sort of got a genotype. So 75% of the SNP were no call, but still we're able with these markers to separate a third-degree relationship to solve it. So even though you don't have a full profile, there may be still a lot of useful information downstreams. So in this interlab study, we also wanted to sort of exercise and analyze interpretation of the SNPs rather than only the genotyping. So of course, if you establish a SNP profile in order to be able to use it also for an application, there are need for tools and reference data for downstream interpretation, sort of at the case level. For example, we had a kinship. We would like to estimate the probability of the two individuals or three individuals tested to be related a specific way compared to being unrelated individuals, or to do ancestry predictions or phenotypic predictions based on the established SNP data. So in this part, which we called part 2 of the interlab study, was focusing on the interpretation. So each lab were given, based on a number of different case questions, were given the same SNP data or SNP genotype, and also some instructions for what to do with it. So for example, the part 2 involved different case scenarios. For example, a kinship case where they were given SNP data to see if two individuals were related as first cousin once removed, or if they were unrelated. Another task that were given was to mimic a DVI case scenario. So SNP profiles were given, also partial SNP profiles for originating from 4 bodies that sort of were believed to come from individuals that disappeared in the 1960s. So for each of the missing data, there were also reference samples or reference data from relatives, or different relatives with complete profile, and the task was given to establish the identity of the found bodies. And also we added some case scenario exercises to do ancestry and phenotype prediction. So here each lab were given SNP profiles from 2 different crime scene samples from unknown donors. And the task were to sort of predict the eye color, hair color, and skin color based on a biogeographical ancestry based on these data and available reference data and tools. So tools were given also and reference data. Each lab were they could use their tool and reference data of their own, but if they didn't have any such, they were given the possibility to use the FamLink 2 software. So this is a software developed mainly by Daniel Kling, which recently also did a lot of effort to also include additional features to this software. And we also added the reference data, thanks, among other, thanks to Chris Phillips for allele frequency estimates for various populations around the world. Overall, the results, I will not go into detail in the outcome, but overall the results from the interlab part 2 were in concordance between the different labs. There were differences. We did see differences when it comes to how the different labs verbally sort of interpreted the numeric outcomes, like the verbal statement. But we will talk about this more and discuss this more in the paper from this interlab study that are coming soon. So to sum this up, there is a need for standardization of SNP markers. SNP markers in forensics have been around for many, many years. However, there is what we believe is still a sort of lack of standardization. And the fourth panel, which is an all-in-one SNP marker panel for different forensic applications, is one such suggestion of a set of forensic SNP markers that can be used, sort of. But there are other panels like the intelligence panel from QIAGEN and other similar panels as well. However, this interlab study showed that it's possible to customize a complete workflow going from DNA to genotype for a 5,000-ish SNP marker panel. In this interlab study, 4 different assays were compared and included, and we did see and can show that across the different assays, all assays did work well. However, there were quite large variability among the different labs due to different factors. And also, the study like this, of course, can only be done through collaborations between different labs, as in this case. So with this, I would like to thank all the participants. It was super fun and important collaboration. We are as we speak, we are writing the paper with all the details and a more thoroughly comparison between the different labs and based on all the data. And it will appear hopefully in a couple of months for you to read. Thanks.